and just talk to me a little bit about gene editing, how things have maybe changed since you started your program, what really excites you about it, a little bit, and just what you think about the landscape
What
overall.
I think about landscape? Oh, I think it's so cool. I came into my program really interested in CAR T cell therapy, and interested in T cells in a whole. I love the idea of essentially exploiting your own immune system in order to fight cancer cells, and it's very different. I think it's one of the emerging pillars in cancer therapy. So then that came, and then okay, CAR T cells, okay, how do we engineer CAR T cells, and sort of like engineering DNA vectors, learning about that and stuff like that. And then, you know, thinking about just the sort of cumbersomeness of CAR T cell engineering, and like learning about like okay, there's a lot of companies that are trying to make the engineering process shorter, the manufacturing process shorter, blah, blah, blah. And then I heard about in vivo cars, and I was like, is that gonna work? Like that doesn't sound. But then you're seeing all these models that are like working, and these companies that are forming and stuff like that. So it's really interesting, and also like how they're changing the vectors that they're using, or like the nanoparticles. Like oh, like everyone knows that lipid nanoparticles really only go to the liver, and then it's like oh well, actually like these nanoparticles are how they're formulating it. Like it can it doesn't transduce hepatocytes or all these things. I think it's just so cool how things are changing, and people are coming up with new ways of engineering these viral vectors, that could then go into gene editing. I'm not, I personally don't like do any of it, but I think it's really like the next big thing.
hmm Are there any other emerging trends or theme in cell therapy research that have stood out to you this year?
Um, I think retrospective studies have really stood out to me, and learning from cell therapy is quite new. So the more patients that receive cell therapy, the more patients that we can learn from. I think a lot of the talks that I've been to have talked about prediction of toxicities, predictions of how a patient may respond. There's so many retrospective studies on like patients that are long term or durable responders, how their products might be different from a patient that did not respond. And I think just the retrospective, looking rather than looking forward, looking back to see how we can make it better going forward. I think that's I like it's great. And also like learning about the new toxicities, because like every like for example the BCMA CAR T cells, they're getting toxicities or neurotox later than what they've seen with like the anti CD19 CAR T cells, and it's like okay, how can we learn from that? Like are we seeing them like in the CSF? Like what's going on there? I think it's really interesting,
Mm-hmm.
and also informs like my work. Like, oh, how can we engineer CAR T cells to maybe combat the things that we're seeing in patients?